Great. So could you walk us through the process of tissue culture?
Okay, sure. I'm going to give you a very brass tacks, very basic explanation of the tissue culture technique. And also, I have some images from
Great.
both our research and also some cartoony ones that are a little bit easier to see if, like, the screen's small, like they're watching it on their phone. So, the basic process of tissue culture is first you do a selection of, of materials that you select a plant and you select the tissues in question, and then you take and we'll pull up this cartoon. So you'll take a cutting from those tissues and sterilize them. This is so that you only are dealing with the plants that you wish to deal with and not of any of the pathogens or any of the other materials. You're only selecting for, like, say, for instance, you only want meristem, which is like bud tissue, or you only want leaf tissue. So you just get that. And then you plate the tissues on a media that has these cell signaling compounds. That's those compounds that I said beforehand were like the plant saying, hey, look, this happened, you need to do this.
Mm.
So it grows into a mass of these undeclared majors of cells, of like undifferentiated tissue called callus. And then you take and you start giving it signals to say, like, hey, I want you to make roots, or hey, I want you to make this compound, or hey, I want you to make shoots. And in this micropropagation slide, what this is doing is we're taking some of the body tissue, converting it into callus, and then converting that tissue in that callus. While we're making the callus grow over and over again, we're also telling it to make more plant babies. Now, that's the cartoon version. These are some of the research pictures. They might not necessarily be as clear. So this is an example where that upper left, the white tissue, that was some cuttings that I took off of a economically threatened plant, and we grew a whole mass of that tissue.
Mm.
Normally, that amount of tissue would take about 2 or 3 years to grow because it's a perennial. This was grown in months, and then we convert those tissues into a special subtype and start making plant babies. That's one example. This is from our lab currently. We're working with some of our partners and doing some work for them. So I took some cuttings off of one of their mother plants, and then then selected for tissues, plated very small tissues. And these guys, what they start doing on that little jelly media that they grow, they're in, they grow, they reproduce, they grow, they reproduce, they get bigger, they reproduce, and more. And then as they get to a certain size, they start throwing shoots off, and then we cut the shoots off and propagate, cut the shoots off, propagate, until you get basic. This is probably a little bit more easy for the audience to see. So that's like the basic process of tissue culture.